Welcome dear viewers to the informative video on stability study protocol. As stability study is one of the most important and critical requirement for the pharmaceutical product filing and approval. Making stability study protocol and having its understanding is very much important, and based on that protocol the stability study is performed and the results are reported. So first protocol is prepared, then stability samples are loaded onto the stability as per the stability study protocol. Testing is then performed and the results are reported. So first stage for stability study is making a stability study protocol. And see, the stability study protocol is a written plan, or it is a document in which there are written instructions, procedures and plan for performing the stability study. Stability protocol is written and it is checked by the professionals from different departments like research, quality control, quality assurance, regulatory affairs and the stability team. Stability protocol is to perform the stability studies, and it is the detailed write-up of all the study information, product information, study requirements, testing and testing intervals, and the packs. So it is to be made as per the regulatory requirements. Also, whenever you are writing the stability study protocol, that time you should also consider the market of the product, whether it is for US region, whether it is for Europe region, whether it is for Brazil region, and so many. So all the regulatory guidelines should be clear to the professionals who are involved in the stability studies. That is very much required. Then three batches are required for the stability studies. As per the regulatory guidelines, minimum number of batches for the stability study for product registration and approval is three batches. Three exhibit batches of each strength are required. The principles of bracketing and matrixing can be done or can be taken into consideration while writing the stability protocol. Bracketing and matrixing design for the stability study I will include in the later upcoming videos. So for now, you remember that bracketing means the extreme conditions are being tested in the stability, like highest count of the HDPE bottle. That means, for example, you are studying the bottle HDPE bottle for stability with the count of 30 tablets and with the count of 1000 tablet. Then you can bracket the in between packs like 90 tablets bottle, 500 tablet bottle, etc. And matrixing. Matrixing means for some of the conditions all the testing is done, and for some of the conditions the testing is not done. That means matrix type of design is done. The batch size is 1 lakh units. For some of the guidelines, 1 lakh units are given as a batch size of exhibit batch. For some of the guidelines, 1 lakh packed units are mentioned. But as a general, always the exhibit batch size is taken around 1.4 or 1.5 lakh tablets or dosage units, so that after getting the yield of around 90% or 95%, you can get the 1 lakh units. Then the pack should be the intended market packs. Whatever the packaging system is proposed for the marketing of that product, that pack is required to be charged onto the stability. And in that pack, the stability is required to be proved. manufactured and packed in the commercial setting with the intended commercial manufacturing process. So these batches are to be done in the commercial manufacturing site. So it is clear now that three batches are required and the packaging system and the manufacturing process should be final. Then what is the information to be included in the protocol? Many of the applicants include much more information. Some applicants use less information in the protocol. For some of the applicants, there is a system of making protocol and getting it approved online, while some of the applicants use the paperwork for making the protocols. But the information should be detailed, like product name, batch number, what was the API batch number used in the manufacturing of that particular batch, what was its expiry and vendor details. Also the packaging material details are required to be included. Then pack style and pack configuration, like blister pack, bottle pack, bulk pack, and the count of blister, count of the tablets or capsules into the HDP bottle. Manufacturing date of the batch. Also, some of the applicants include tentative expiry date or proposed expiry date. Then batch size, protocol number, label claim of the formulation, and market details. Many of the time it happens that the product is for global market, like it will go to, or it may go to US, Europe, or CIS countries, or the Russian countries, or the different climatic zones like zone 4B, like Brazil market is for zone 4B. So like that, the market details should be included. And what is the reason for study is to be stated, that it is the new product stability study for filing. Is it a SUPAC change into the formulation, and that's why the stability is going to be run? So this information will give understanding and clarity to the professionals who are working or dealing with these things. Stability in date and the completion date. Stability study interval and completion conclusions. So always there should be a space to write the conclusions, so that all the information will be at one place and in one protocol. Now, what are the type of study can be done in the same protocol? So why I am saying that? Because some studies are done at the similar conditions in the similar packaging. So these studies can be bracketed or can be taken into one formal stability protocol, like formal stability study to assign the shelf life and expiry period, that is known as formal stability study. Also, the formal stability study is done to have the storage recommendations. Then you can include the in-use stability study in the same protocol, and at initial and the last out time for the samples, like for first month you load the initial samples and perform in-use stability. After 1 month or 2 months as per the product requirement, and one sample you load for the last time point for the long term. So around 23rd month you can take out that in-use stability sample and perform the in-use stability. So like that, in-use stability can be made in line with the formal stability study. Then polymorphism or polymorphic study can be done in the formal stability study. Polymorph conversion or retention, whether the polymorph of the drug get converted into other form or whether there is no change. Sometimes the amorphous form of the API may convert to the crystalline form. So for that the polymorphism study is performed. And these studies can be included in the same or single formal stability study, and that will be advantageous. Stability conditions and the testing frequency. it is very important to have the clear understanding about the stability conditions and what are the frequencies. So conditions are determined based on the market requirement for the formulation, based on the regulatory requirements and guidelines, and also the stability zones or climatic zones. So zone 1, 2, 3, 4A and 4B. So you first of all you should have the information where the product is going to be launched or going to be marketed, and based on zones stability studies can be performed. Generally the long term condition, intermediate condition and accelerated condition is the condition for which the stability study is performed. The conditions are as per the regulatory requirements, climatic zones and the intended market country for the specific products. Long term condition is 25 degrees Celsius and 60% RH. Sometimes the long term condition is 30 degrees Celsius and 75% RH. Intermediate condition is 30 degrees Celsius and 65% RH. And accelerated condition is the same, that is 40 degrees Celsius and 75% RH. So if you consider the climatic zones, then you can have understanding that zone 4B requires the long term condition of 30 degrees Celsius and 75% RH. Zone 4A is 30 degree and 65% RH. Zone 3 is 30 degree 35% RH. Zone 2 is 25 degrees Celsius 60% RH. And zone 1 is 21 degree 45% RH. Generally, the long term condition is 25 degree 60% RH and 30 degree 75% RH. So in general these conditions are included, and if you consider the two long term conditions like 25 degree and 30 degree, then you have to perform the stability study with four conditions: two long term condition, one intermediate condition and one accelerated condition. So long term condition has the highest testing frequency, or the time points are more, like 3 months, 6 months, 9 months, 12 months, that is 1 year. And after that 6-month interval will be there, like 18 months, 24 months, and after 2 years 1 year gap will be there. That is after 24 hours directly it will be 36 months. And after 36 months you can also go on for the long term studies if the longer shelf life is required, or if the extension to the shelf life is required, and that information is also captured into the protocol. Intermediate condition is 3065 and it is with the testing frequency of 3 months, 6 months, 9 months and 12 months. So intermediate condition run for 1 year. Accelerated condition run for 6 months. It is 3 months and the 6 months. The intermediate condition testing is done if the accelerated condition gives the failure results for the product, or if the product fails at the accelerated conditions. Now samples. So how much sample are required to be loaded? As a general criteria, the sample should be sufficient for analysis for 2 to 3 times. So always load the sufficient sample, so that if there is any problem in the analysis, then you can take out the fresh sample and do the analysis. Also you can do the investigation if required. Then sample for polymorphism study, samples for in-use study, placebo samples and additional samples if required. So always think to load sufficient sample. The initial test is performed on finished product and data available in the COA can be utilized as a initial data. Testing of intermediate condition is done if the product fails or if any significant change is observed at accelerated storage conditions. If the product is not stable at higher temperature, then the note can be given into the protocol that the product is not stable at accelerated condition, but still accelerated condition is required to be performed, and if it fails at that accelerated condition, then directly intermediate condition is required and it is required to be tested at intermediate condition, so that the people working on this stability study will have the clear information. Now, what are the tests? So, tests are physical testing, chemical testing, polymorphism testing, and the microbial testing. So, physical tests are like appearance or hardness, disintegration. These are some of the physical tests. Physical testing of any change in the color, change in the shade of tablet, or change in the capsule color. So, these are the physical tests. Chemical tests are done at all the time points for all the conditions in all the packs. There is no skip for this chemical testing. Description, assay, dissolution, residual related substances or organic impurities, water content, LOD, disintegration testing, hardness are the main tests in the chemical testing. Also, the specific impurities can be tested like chiral impurities or enantiomeric impurities or any impurity which is to be monitored. Then polymorphism testing is performed for long-term and accelerated condition as per the product requirement. Generally, it is not done for the bulk packs because bulk packs are not going into the market, and also these bulk packs are packed for the remaining samples after the stability packaging. Microbial testing is done for long-term conditions and generally it is not done at accelerated condition because at accelerated condition the humidity levels are higher. Also the temperature levels are higher. Microbial testing are done at 12 months, 24 months and 36 months for long-term conditions. Then the packs. What are the packs? For if you take example of tablet, so you will have different packs like alu blister pack, PVC blister pack, PVC PVDC, desiccated foil pack and many more. Bottle packs like HDP bottle pack with different counts with or without desiccants. Desiccant like silica gel, molecular sieve, oxygen absorber, activated charcoals. These can be included as per the product requirement. Then bulk pack may be HDP bottles with 1000 count or 500 count or more, and alu pouch or triple laminated foil pouch or pack for the bulk packaging. So these are some of the packs. Stability study is a big topic, it is a vast topic and it cannot be covered in 3-4 videos. I have made different videos for different aspects and this is a video for protocol. So understand the protocol writing because the protocol will serve as a base document for the entire stability study. I have referred ICH guidelines for making this video. Also, I have referred ANDA stability testing for drug substances and product guideline from USFDA and the EMA guideline on stability testing. I hope you might have got good information about stability protocol and stability study from this video. So keep watching the Pharma Learning in Depth channel. Also watch the other videos which are already present in the playlist of Pharma Learning in Depth channel, as these videos will help you to understand the terminologies. Thank you for watching the video. Please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.